Hello, my name is Sarah Shuachea, and today I will be presenting my science fair project called Unraveling Protein Complex Mysteries. My science fair project is about new therapeutic modalities in the world of drug discovery and the growing capabilities of artificial intelligence. I wanted to conduct a science fair project that was new to me and harmonized medical advancements and artificial intelligence. I would like to begin by explaining the components of my experiment. Let's start with the ubiquitin-proteasomal system. The ubiquitination system, or the ubiquitin-proteasomal system, is a natural process of degradation of dysfunctional or diseased proteins, often known as target proteins or proteins of interest. Three enzymes, E1, E2, and E3, work together to attach a ubiquitin chain to the protein of interest. This signals for the protein's degradation in the proteasome. A new therapeutic modality, the proteolysis-targeting chimera, otherwise known as a PROTAC, facilitates the ubiquitin-proteasomal system. A PROTAC consists of three parts, a linker and two ligands, one connecting to the target protein and the other connecting to the E3 ligase. This connection, made by the PROTAC, strengthens the bond between the E3 and POI, allowing for the facilitation of natural degradation. Here you can see more clearly how the PROTAC connects to the E3 and the target protein. The fact that it connects to both the E3 and the protein of interest makes the PROTACs heterobifunctional. PROTACs are a relatively new invention, but they pose several advantages to traditional small molecules. Lower dosage is required as the PROTAC can remove itself before the target protein gets degraded by the proteasome. This allows for the PROTAC to degrade many target proteins, meaning it can work catalytically instead of stoichiometrically, as in only degrading one protein. Additionally, what was once known as undruggable proteins are now druggable by PROTACs, as they do not require to bind to an active site to degrade a protein. For the same reason of not needing to bind to an active site, resistance mutations are less likely to form. Due to the fact of high specificity, as the heterobifunctional PROTAC must bind to both the E3 and the POI, there are less likely to be side effects. The algorithm used in the experiment is called AlphaFold Multimer, which is an artificial intelligence-based algorithm for predicting the interfaces of protein complexes. To do this, AlphaFold Multimer must be provided with an amino acid sequence, which is then inputted into multiple sequence alignment, or MSA. This is a technique that identifies and aligns homologous protein sequences. Evoformer then perceives and recognizes patterns and relationships within protein sequences to determine the most suitable template for prediction. The protein structures are then manipulated to create the 3D structure, and the patterns determined by artificial intelligence based on the PDB training set ultimately determine the structure and arrangements of the protein chains in the complex. Finally, the details are refined. The algorithm provides the top 5 predictions, which are ranked based on confidence. All this data of the predictions and confidence graphs is downloaded. While it is known that AlphaFold Multimer's resulting individual protein structures are highly accurate, its accuracy in predicting the relationships of protein complexes is not as widely known. By using AlphaFold Multimer, its accuracy in predicting protein-protein interfaces and its capabilities in advancing the PROTAC discovery can be found. Therefore, the problem statement of how accurate AlphaFold Multimer is in predicting the interfaces of protein complexes composed of a target protein and E3 that corresponds to the same experimentally determined ternary complex, including the PROTAC, is posed. Due to the uncertainty in the variety of complexes in AlphaFold Multimer's training data, the hypothesis, if the E3 ligase and POI are similar to reported TBD ternary complexes, then AlphaFold Multimer will be able to predict the quaternary structure of the protein complex with high accuracy is posed. Next, I will go through the procedures taken to conduct this experiment. First, the protein data bank, otherwise known as the PDB, was searched for ternary complexes consisting of an E3, POI, and PROTAC, which are named by PDB IDs such as 8C13 as shown on the screen. Possible ternary complexes were downloaded onto an Excel file where the information of each ternary complex was checked to see if the complexes consisted of an E3, POI, and PROTAC. The primary amino acid sequences of the E3 and POI were then inputted into AlphaFold Multimer, more specifically CoLabFold v1.5.2 patch, AlphaFold 2 using MMSec2. Once the prediction was run and the data was downloaded, a DockQ score was used to evaluate the prediction's accuracy by comparing the difference in atom position between the experimentally derived ternary complex from the PDB and the prediction from AlphaFold Multimer. The graph here shows what would be considered an inaccurate, acceptable, medium, and high DockQ score. AlphaFold Multimer's confidence graphs are shown here. The top left and the bottom right of each square shows its confidence in its individual protein complexes, mainly shown in blue, meaning that it has high confidence. Though the top right and the bottom left, which are the squares for the protein-protein interfaces, are mostly depicted in red, showing low confidence. This is for all except the square for rank 1 of the protein complex corresponding to the PDB ID 7KHH. In this maestro visualization, AlphaFold Multimer's 5 ranked protein complex predictions are depicted. Panel A and B show the PDB ternary complex with and without the PROTAC. Panels C through G show the 5 predictions from AlphaFold Multimer. In panel H, it can be clearly seen how all the protein complexes are arranged differently. Though if you look closely, it can be noticed that the individual protein structures are the same, but just arranged and shown differently. The data shows that AlphaFold Multimer had an extremely low DockQ score due to mistakes in the protein-protein interface, though there are some outliers with acceptable DockQ scores. This graph shows how the majority of the predictions had low DockQ scores, but some were acceptable. This maestro visualization further supports that the arrangement of the individual protein complexes resulted in the corresponding DockQ scores. Panels A through D show predictions with acceptable DockQ scores, as the gray almost aligns with the blue, though panel E and F show predictions with low DockQ scores where the arrangement is visibly inaccurate. To compare AlphaFold Multimer to another algorithm, DockQ scores for PRosettaC were obtained. PRosettaC is a physics-based algorithm compared to AlphaFold Multimer's artificial intelligence-based algorithm. PRosettaC first samples the distance between the two ligand anchor points. PatchDock, a global docking algorithm, then inputs the constraints of the distance. Possible models are then made with RosettaDock, a local docking algorithm which ignores the distance constraints. The final ternary complex is made by incorporating both global and local docking. The ternary complexes are then scored and clustered based on the score. PRosettaC produced more accurate DockQ scores than AlphaFold Multimer. As shown in the graph, PRosettaC's overall mean and outliers derived from its resulting predictions are higher than AlphaFold Multimer's. The reason AlphaFold Multimer did not produce predictions with high accuracy was due to the absence of PROTAC information, resulting in the algorithm wanting to find a direct link between the E3 and the protein of interest. Multimer also had a preference for the lowest binding free energy conformation, which was learned from the majority of the training data, though this does not correspond to the experimentally derived ternary complexes. Meanwhile, PRosettaC produced more accurate predictions, as not only is the algorithm built specifically for predicting ternary complexes consisting of an E3, POI, and PROTAC, but the PROTAC information must be provided. PRosettaC must also be provided with a file with the individual protein structure information, as it cannot build the protein structure from its amino acid sequence. AlphaFold Multimer's methodology of using artificial intelligence rather than physics makes its algorithm slightly more notable. While AlphaFold Multimer did not provide accurate predictions, by establishing a baseline of its capabilities, its limitations can be addressed. Resultantly, better AI algorithms can help advance PROTACs even farther, as well as help utilize untapped E3 ligases and establish common E3s even more. And lastly, conducting an open source study and sharing the results holds promise for advancements in the broad science field in general. I would like to acknowledge my mentors, Professor Stefan Schuacher and Joey Schultz, as well as my teachers, Dr. G, Miss Usatores, and Miss Valdez, for helping me along the way. And thank you to the judges of the science fair and to those who have listened to my project. I hope you enjoyed learning about my science fair project. Thank you again.